Hello, everyone. I'm Eric Isaacs, president of the Carnegie Institution for Science, and I'm delighted to welcome you today as we join the Kavli Foundation and their new president, Cynthia Friend, the Norwegian Embassy in the United States, and the Norwegian Academy of Sciences and Letters in presenting this Kavli Prize Laureate lecture. Our featured speaker today is Stefan, who won the Kavli Prize in Nanoscience in 2014, and the same year he was awarded the Nobel Prize in Chemistry. These awards recognize Dr. Hell's revolutionary work in microscopy. For most of the 20th century, it was accepted, it was well understood even, that an optical microscope could not resolve or see features or objects smaller than about 200 nanometers, which is about half the wavelength of visible light. And at that point, at 200 nanometers, or half the wavelength of light, the image is no longer constrained by the quality of the instrument, but by the wavelength of light itself. But then in the 1990s, Dr. Hell developed a new method of microscopy based on fluorescence emissions that made it possible to start achieving resolutions of about 20 or 30 nanometers, which, if you think about it in the number of atoms, that's just tens of atoms across, maybe 30, 40 atoms across. So quite exquisite resolution. Remember, with a wavelength that's 10 times or 20 times larger. And more recently, he has been pioneering a new method that I think we'll hear about that can use visible light to achieve resolution at the molecular scale, just a few nanometers in size. So this level of resolution is really, it's fantastic. It's really revolutionary and crucially important, in particular in life sciences. In many things, we can use X-ray microscopy, which has very, very short wavelengths, to make observations at the nanoscale, even the atomic scale, but that's not possible with live organisms. With optical microscopes, such as what Professor Hell has developed, it won't kill the organism. You can actually watch the organism in vitro, in its live state. It is just tickled a little bit by the light. So Dr. Hell's fluorescence microscopy has made it possible to make direct observations of dynamical processes in living cells with this kind of nanoscale resolution that has the potential, has the potential to transform our understanding of the way life works and disease. Dr. Hell comes to us today from Germany, where he serves as director of the Department for Optical Nanoscopy at the Max Planck Institute for Medical Research and the director of the Department of Nanophotonics at the Max Planck Institute for Biophysical Chemistry. He first joined Max Planck Institute in 1997 after holding research positions in the European Molecular Biological Lab and EMBL at the University of Turku in Finland and the University of Oxford in the UK. He holds a doctorate in physics from the University of Heidelberg. Today, we're also pleased to welcome back Frank Sesno, who has led a number of these Kavli conversations. Frank is the Director of Strategic Initiatives at George Washington University and the creator of Planet Forward, a web and televised project that highlights innovations in sustainability. Before coming to GW, George Washington University, he spent 20 years at CNN, where he served as the White House correspondent, anchor, and Washington bureau chief. And like you, I'm looking forward to a fascinating conversation with a brilliant researcher who literally changed the way we see the world. So please join me in welcoming Dr. Stefan Hell and Frank Sesno.
Thank you very much, Eric. Really appreciate that. And Stefan, good to see you. Thank you for joining us. I can't hear you, though. Perhaps you could unmute yourself, and that will work better.
Hello, Frank. Hello, everyone.
There we go. Well, we'll get to a few things, and I know you have some slides, and we're going to illustrate some of your conversation. But since this session is called the Resolution Revolution, I think I should start by asking you, do you consider yourself a revolutionary?
I didn't think about it that way, but yes, I try to do something that makes a difference. If it turns out to be a revolution, the better it is, of course, but I didn't think about it that way initially.
Well, I think as Eric indicated, it really has been a revolution. I want to share with our audience that you've got a great treat ahead of you in listening to Stefan Hell here. Not only is he a revolutionary, but he's also a wonderful storyteller. And I'll just give you a little hint here. Does this resonate with you for some reason?
Oh yeah. It's a donut, obviously.
We'll come to that, and we'll have an opportunity to talk about donuts. But let's get to your science and to your work. We'll incorporate your very dramatic life, which has informed your science, your work, your risk-taking throughout. We'll also have an opportunity to have questions from the audience, and I'll remind the audience, please feel free to put your questions in the Q&A function here, and we'll get to those later. But Stefan, start by telling us about the problem that you first zeroed in on, and why that problem fascinated you.
Right. Yeah, so I'm sharing my slides now, and I hope you can see them. So, you know, I mean, visual perception is really, really important for mankind. And of course, it's very, very important for science as well, because, I mean, this is the only way to see small things. And this is why actually the natural sciences started out with crucial developments in microscopy. And light microscopy was key to finding out that basically every living being consists of cells as basic units of structure and function. Of course, with light microscopy, subcellular organisms organelles were discovered, like the mitochondrion. So light microscopy was incredibly important for the natural sciences in general. However, I'm sure you will remember your school days when you learned that the resolution of a light microscope, as Eric just mentioned, is fundamentally limited by the wave nature of light. So you cannot discern features that are closer together than about 200 nanometers. So that's a fifth of a thousandth of a millimeter. So that's almost nothing. I mean, and of course, the eye couldn't tell distances of that scale apart. And this is why scientists developed electron microscopy in the 20th century. And electron microscopy, because it relied on electrons rather than on light, could achieve a much higher spatial resolution, in fact, down to the molecular scale or to the atomic scale. However, what electron microscopy could not do, it could not visualize living cells or living matter, only that material. And so there was a need actually for a method that could see living cells or could look into the cell and see, for example, proteins or other biomolecules with a very high specificity. And that was also not really possible in electron microscopy. And therefore, it was clear that if somebody came up with an idea that overcame the diffraction barrier, then it would make a big difference in the life sciences. And so this was a problem that I actually worked on. But to be honest with you, when I started looking into this problem, I didn't set out to do something good for humankind or so. I was just interested in the problem, you know.
Well, it turned out very well for humankind. You were up against what you called the limits of resolution, right? So what was the problem specifically, and why was it so hard to break that resolution barrier?
Yeah, it can be explained in very simple terms. I mean, a light microscopy or a light microscope, basically, or the most important element of a light microscope is the lens. It's just a piece of glass that is shaped in a particular way so that it focuses the light down to a point. But because light propagates as a wave, and usually nowadays lasers are taken as light sources, because light propagates as a wave, it's not possible for the lens to concentrate the light on a point, on a geometric point, literally on a point. For example, let's assume this is a sample and these are points, the points are molecules. It's not possible to concentrate the light on a point, because then you could, of course, discern the molecules if you could do that. What will happen instead is, because of the wave nature of light, we will get a spot of light that has a finite extent. And this spot of light is at least about 200 nanometers across and about 1 micron, so about 1,000 nanometers along this axis. And therefore, all the molecules that happen to be located within this spot of light, they will be illuminated at the same time, simultaneously. There's no way out of that. And therefore, they will also scatter light back at the same time. So the light will come back at the same time. And so, so each of the molecules, of course, will kind of produce a blob of light here at the back end at the detector. And the signal from each of the molecules, of course, will be confused here at the end, because all of them emit light at the same time, and no detector could ever tell which of the molecules was the actual source of light. So the light will be confused, and this is why it's not possible to tell the signal from these molecules apart. And no matter what you placed here, the eye or even a sophisticated camera or whatever, It's not possible for any detector to tell those molecules apart. Now, this problem was realized by a person who lived at the end of the 19th century here in Germany, actually graduated here in this town, Ernst Abbe, and he kind of formulated this problem in an equation that is named after him. This is Abbe's equation. And it's basically saying that features that are closer together than the wavelengths, lambda, divided by this number, which is the so-called numerical aperture of the objective lens, cannot be told apart, simply because they fall within the same spot of light. And this is a number like 2 or 3, and the wavelength is something like 500, 600, and this is why you end up with 200 nanometers in here. And that has been the barrier, so to speak, for many, many, many years.
And that's what I mean, that was commonly believed, right? That was,
yes.
Everyone believed that.
Everyone believed that. So that you find this equation basically in any textbook of physics, of course, also cell biology and molecular biology textbooks, because of the relevance of light microscopy to these fields. And not only that, I mean, if you will, you can also find it on a memorial that was erected in honor of Ernst Abbe in Jena. This is the town where he lived and worked. And there, his equation was literally written in stone.
Carved in stone.
Exactly, carved in stone. And this is what scientists really believed throughout the 20th century. Not only they believed it, this was also a fact, because if you took a picture, say at the end of the 20th century, the resolution was like this. So this is actually a filament of or filaments from a cell, and you can see that the filaments look blurred. And so this was the end of the story. I mean, you couldn't get any better than that. Now, the development that I'm responsible for, STAD microscopy, that was just mentioned initially, showed that that's not true. This limit can be overcome. So there is physics in the world that allows you to get sharper pictures. And this was kind of not expected, because everyone thought that would be impossible, because it was kind of against the law that seemed to stand forever.
Did you walk around with this vision, this thought in your head? Did this come from some, bolt of lightning? Or, I mean, how did you end up working on this problem?
Yeah, it's a very interesting story, as a matter of fact. I mean, it's not that it struck me all of a sudden. It's actually tightly connected with my life. And I would like to show you actually how I got there. And it was not a straightforward, say, way towards the goal like that. It was rather meandering and coincidences and ideas, of course. Now, I was born in Western Romania, actually, and in 1962. And of course, you will still remember those days. This was the time during communism. This was the time of the Iron Curtain. And it was Ceausescu communist Romania. And of course, most people felt at that time, this is a system that is not really allowing people to develop the way they should and do the best out of their lives. And apart from that, I was part of, or my family was part of a German ethnic minority. And so we took advantage of that fact, and realizing that there would be actually no real future in this country, we took advantage of the fact to get out of the country and to move to West Germany. And so after some problems, we succeeded doing that. Actually convinced my parents at the age of 13 to try to apply and get out. And we managed to leave Eastern Europe, Iron Curtain, cross the Iron Curtain, and settle in this town called Ludwigshafen, in West Germany at that time. And we didn't have any relations there, actually. And you may ask, so why did we settle there? It's very simple. My mother was worried that there would be a clash between East and West. And she said, okay, we have to go somewhere west of the Rhine, because if there is a clash between East and West, the Americans would not allow the Soviets to cross the Rhine. And this is why we ended up there. And the reason why I liked this place was that it was close to Heidelberg. And Heidelberg is a university town, and I knew that, okay, this is a good place, and it's good to aspire to get educated there. And so after getting my high school leaving certificate, I studied physics in Heidelberg. And like most physics students at that time, I was very much attracted to, say fundamental physics, like studying, so basic forces and basic interactions, of course, particle physics and cosmology and things like that. But at the time, I had to make a decision on what to study. When I had to choose a subject, actually, for my PhD, and this is what I looked like back then, believe it or not, I felt that I shouldn't do that, because I spoke to other students and they said, don't do that because there is a surplus of physicists. And if you do something that is very theoretical, cosmology or something like that, you will end up as a taxi cab driver or something. You will end up unemployed. And of course, this made a lasting impression on me, because my parents still had to struggle to settle in the West. My father became unemployed for a short time, and my mother got a serious disease. And so I felt I have to take care of my parents. I have to do something that is very applied. And so that gets me a job eventually with a company. And so I signed up with a professor, who was actually became my thesis advisor, who started a startup company. And the startup company specialized in developing light microscopes for computer chips. And I felt, okay, if you do a job in his lab, of course you qualify for a job with IBM or something. And so my job was actually to kind of investigate how well you can see tiny little structures on computer chips with the light microscope. And I felt at some point, this is totally boring. This is not why I studied physics. I was interested in basic physics and not in this, say, boring microscopy, boring subject of the 19th century. And to be honest with you, I was just about to become miserable. I was so frustrated because I felt that I'm doing something that I actually don't want to do. And so out of this frustration, I thought maybe there's still an interesting problem left that could be worthwhile pursuing in this, say, boring subject of, say, light microscopy, 19th century physics subject. And I thought, okay, the only thing worthwhile pursuing would be breaking the diffraction barrier, because that's something no one believes is possible, and that would be kind of cool and basic and interesting. I didn't think about the implications it would have for the life science to do something good for mankind or whatever. I found it cool. This was the only cool problem left in this boring subject of light microscopy. And this is how I got actually into the field.
Well, I think we're very glad you got bored. We're very glad you pursued light microscopy in this way. I think I'm most impressed by the fact that at 13 years old, you convinced your parents to move west. So that worked. Now, this is where the donut comes in, I think. And it's, props are good. So let you take it from there. What was your fundamental insight and what's the donut connection?
Yeah. Actually, so then after I realized that that could be a way to go, it would be worthwhile pursuing actually this breaking of the diffraction barrier. I tried to convince people in Heidelberg, at the University of Heidelberg, my thesis advisor and other scientists, to go after the problem, but they didn't want to be associated with the thing. And so I actually really ended up unemployed for a short period of time. But I had learned the hard way that it's better to do something that you want to do, that you like, rather than doing something out of the thought that you need to have some sort of, say, social, say, security or safety or something. And so I decided to take a risk. And so I couldn't find a job in Germany, but I was very fortunate that I met a person who started a lab in Finland. He was a Finnish person. He returned back to Finland. He said, why don't you come to our place, to Turku? That was a city I had never heard of at that time. And maybe after a while, you'll find a way and you'll be successful. And as you're saying, indeed, about 6 to 8 weeks after I got there, I had this decisive idea, and I'm going to show you now what it is. And indeed, it has something to do with a donut. Now, as I explained, the problem was that all the molecules here produce light at the same time because we have this blob of light. So I realized there is no way to change that. You cannot make that light spot smaller, no way. But what you could do is you could make sure that not all the molecules that are covered here by the green light are in the end capable of emitting light back to the detector. So what I did is I introduced a beam of light that turns molecules off. rather than on. So it's a red beam of light that has a certain wavelength that is designed to turn molecules off. And now, of course, you don't want to turn all of them off because then you would see nothing. That's very clear. So what did I do? I realized there is donuts, and not only you can knead them, you can also make, of course, beams shaped into donuts. They're really called donut mode in physics. And so I took such a donut-shaped beam and used it to turn those molecules off. So now you see the red light is used in such a way that it turns those molecules off. And you see only that molecule in the center is now left over. And now the whole system behaves as though only this molecule would have been addressed. And now you see the signal only from this molecule. The confusion here at the detector disappears because all these molecules are, so to speak, silent, remain silent. And then we can get a signal from this one, we move on to the next, move on to the next, and so on. And then we can take a picture with high resolution. That was the basic idea.
What's happened since you got your Kavli Prize in 2014 with this technology and your work and research?
Yeah. Well, you may think that, well, actually, I published it together with an intern who worked with me at that time in Finland in '94. And you may think, okay, now people said, great idea, go ahead, do it, and you get the money. As I said, I didn't really have a lab at that time. The problem was that no one believed this idea. Even though it was published theoretically, it was not an experimental proof at that time. So it took me about 3 years. I lived on scholarships and so on, and tried to convince people in Europe, in the States, somewhere to give me a.
What do you mean? What do you mean? No, what do you, if I may, Joe, because you said something so important there, and I really want people to realize this. When you say nobody believed this,
yeah, because
is that literally true?
Yeah, literally, because it was so outlandish, put it that way, so strange. I mean, overcoming the diffraction barrier. I mean, of course, I had arguments, physical arguments, but people would say, yes, I see your points, but it will not work because of this, because of that, because of that, because of that. Basically, they found arguments why this theory is not viable in the end. And also, I mean, I didn't have the lab to do it. It could have been done in other places, in other labs, in Europe, in the States, in Japan, but people didn't do it because they felt it's not viable. I think one of the reasons was also that in the 20th century, many people claimed that they would be able to overcome the diffraction barrier, but they didn't deliver. And so it was very weird that there's a guy in a place no one has heard of claiming that he could overcome the diffraction barrier. And then why should that be viable all of a sudden? I mean, and after all, it was textbook knowledge that this is not really viable. But I was very fortunate, actually. I happened to meet a person who was at that time in charge of the institute where I'm right now. He's actually American. He was trained in Johns Hopkins, Caltech, and he at that time ran this institute. And he said to me, why don't you come to our place and we give you some time, 5 years time, and then you get some money, and then we'll see if you're right or not. I mean, let's give it a try. Unless you do it, we won't know. And it turned out to be viable. And you see here before and after. And then it was clear that the physics is there and it really works the way I had predicted. So the clear improvement the improvement in resolution became apparent. And you see it, of course, here in certain protein complexes. These are little openings in the envelope of a cell nucleus. This is before and this is after. And obviously, the increase of spatial resolution here, roughly by a factor of 10, is quite apparent. It's very nice to see this 8-fold symmetry in the so-called nuclear pores. And if you compare it with the standard resolution, at that time, you see nothing here. By the way, these pictures, the very same pictures are now in textbooks, in high school textbooks in this country, but I still like to compare the two pictures because I think I still enjoy seeing the comparison, I must admit, even though it's now textbook knowledge.
I mean, it's really incredible, and you can, as you say, you can see protein tubes in a cell wall. You can decode chromosome distribution in a bacterium, or look at how a nerve cell is constructed. So,
right.
How does this molecule scale resolution bear on the life sciences? Because this is a vital tool.
I mean, history has shown that if you see things in greater detail, you get more information.
Sure.
And of course, electron microscopy has been around, but you cannot see things, you cannot see certain proteins highly specifically, because in order to see a protein specifically, you have to highlight it, because there are about 10,000 different proteins in a cell. And this highlighting procedure is very hard to do in electron microscopy. Here it's possible. There is a certain protein that has been labeled, say, with red color, and this is why I can see it. And a certain protein has been labeled with green color. And so I can see how they relate in space. And that's hardly possible, or virtually impossible in electron microscopy. And because you can map out now the proteins in a cell, you can get a better picture of how the proteins are arranged at each point in time in the cell cycle. And this is the actual benefit. So to give you an idea, so 80% of the cases when a life scientist just goes to a microscope to ask a question, 80% of the cases it's a fluorescence microscope and not an electron microscope.
It's really remarkable, really remarkable. Where is this all going, and this scale?
Yeah. So this has been a kind of, I mean, it changed the mindset of people. I mean, it showed that, okay, there is a way to go. And theoretically, at the time I got the idea, back in the 90s, back in Finland, I realized that in theory, the limit would be the molecular scale. So the paper is even saying in principle resolution can be infinitely large, meaning that at the size of the molecule. But we weren't there at that time, not even at the time the Kavli Prize was given. So we had only the factor of 8 to 10, so what you've seen. Now, so my people and I here in the lab have taken it further since then. And we've developed a method that is kind of related with this initial method called stat microscopy. It also uses a donut. You see, the donut is critically important.
A different kind of donut here.
Yeah, and it's a glazed green donut. and so what we also say took actually as a basic element is a feature that was first done by an American colleague, Eric Betzig, turn molecules on and off individually, chemically, rather than say in a bulk as it was done before. And the combination of the two elements actually led us to extremely high spatial resolution. So now, rather than turning molecules off in here, we leave them off, turn just one on, and then the only thing that is left is to find out where it is, because the molecules are turned on stochastically in space somewhere. And guess what? We use a donut beam. Now, green light to turn on molecules, make them glow, to make them emit. And then, of course, we need to find out where it is. And a very, very effective way of finding out where the molecule is to minimize actually the fluorescence or the emission of the molecule, the fluorescence emission, so the signal that is produced here at the detector, by moving the donut center just over the position of the molecule. So by making the two coincide in space, the center of the donut, the whole of the donut, with the molecule, we know exactly where the molecule is, because when the two coincide in space, there's no signal at the detector. Now there is your signal, you see, because there is some light. But once you have matched, so to speak, once you have matched the donut actually with the molecule like this, then you can be sure that that you know where it is, and this gives an extremely high precision. And the separation is done by the on-off transition of the molecule. And this has now led, this is now called min flux. This related concept has now led to very high spatial resolutions, as it is shown in here. Again, these are nuclear pores, but now you see the level of detail is even higher. So it's now increased actually by a factor of 10 over what was possible before, and as you can easily figure out, by a factor of 100 over the initial situation that we had in the 20th century all the time. So this is now the state of the art, if you will. So the first method brought us a factor of 10. Now we have not a factor of 10, a factor of 100. We are down basically at the molecular scale, so resolution of a few nanometers.
It's amazing, and you're moving into production with these things too, right?
Yeah, I think this is important, because in the end, I mean, it's a tool. I mean, what we do is what we have developed is a tool. Of course, the way to get there has been a scientific journey, no doubt, driven out of curiosity and not with the goal of doing something, say, profound or so, but it's useful. And of course, I'm just showing you one example. It's being used for studying proteins in a synapse. So this is it's a very high resolution 3D image of a certain protein called PSD95. That's a very, very important in a synapse. Or you can also use it for tracking movement of molecules in a cell membrane of lipids, for example, with very high speed, because not only the method provides a high spatial resolution, also provides a high speed for a given resolution. And so there's many opportunities that are out there. As you're saying, in order to disseminate the technology and to make it available to the greater life science community, I've set up a company with my students, with my former students, and they have now kind of developed a system that can be used by biologists.
It's fabulous. Let's range over some other issues here, and then I want to get to some questions from the audience very soon. And perhaps we can drop the slides, Stefan, because I know you're running those, and people can see us in all of our all of our glory here. By the way, you're in your office now, right? Where are you now?
So I'm in my office in the Max Planck Institute for Biophysical Chemistry in Göttingen. That's right in the geographical center of Germany, so halfway between Frankfurt and Berlin.
I want you to, because I think this is so dramatic, and the audience, our audience would so appreciate this, to talk a little bit more about your personal journey, because in some of the conversations you and I have had, I was so impressed by the connection that you make between your journey from Romania to the West, the transition that you went through intellectually, the politics that you saw in these different places, and your own personal and sort of scientific interest in taking risks. What is the connection there?
Yeah, I mean, I think one thing that kind of had a strong influence on me was the fact that in communism, of course, everyone or most people realized it is not going to work, and it was very obvious that what you could hear in the news, was not true. And this kind of feeling actually that public assertions or something that is repeated on TV all day may not be true resonated with me, and it may have had an influence, psychological influence on me saying challenging accepted perceptions. So when I heard about this Abbe diffraction, I say, okay, everyone is saying that, but is that true? I grew up actually with this feeling that publicly asserted things may not be entirely true, and it may have had an influence on me, at least psychologically, to venture into something that or challenge something that people believe it is true, but in fact, if you look at it very carefully, it's not.
We have some very good questions from the audience, so let me go to some of those now, and we'll weave other things in as we go. But Jonathan asks, is there some variant of this technique that can help telescopes surpass the diffraction limit?
It's a very good question. The answer is, I doubt it. And I think the answer is no, because in the end, a key element of the improvement of resolution is that we turn on and off molecules. I don't think you can turn on and off stars. If you could do that, if you could do that, you could sort it out. Of course, there are situations where a star disappears for some reason. Of course, then this is being used for separation. But the problem here is, or a fundamental solution to the problem is that we play with the states of the molecules. We manipulate the states of the molecules. And this was not done 20 years back. People didn't realize that the way to go is to manipulate the states of the molecule. At least they didn't know how to do it. Right.
So to that point, there's a sort of related question from Richard, who turns it in another direction. He asks, has this technique been applied to plant cells with chloroplasts, where the chlorophyll is itself fluorescent? Can you see individual chlorophyll molecules?
So we have not, personally, I didn't apply it to plant cells. I know that there are scientists interested in that. So I'm not so much of a person who applies things, because I'm a physicist, of course, who tries to find solutions for physical problems. But I know that there are people who look into plant cells with either STED or with this min-flux method. That's right.
John has a question. How practical / expensive, he asks, is this technology? For example, could museums use it in educational programs as they do with electron microscopy?
It can be done. Actually, as a matter of fact, it has been done. Say the STED method has been kind of turned into a say museum's device that actually here in town, and that is possible. The other method eventually can also be turned into something that can be used in a museum, but it's not so easy to see actually the single molecules going on and off. The other method is the first method I talked about. Yes, it has been there has been a kind of mock system that kind of produces these images under say museum conditions, so that somebody can come, a child could play and get a feeling on how it works.
Pretty amazing. Pretty amazing. Pretty inspiring. If we want to inspire the next generation of scientists, let's do that. Let's put that tool in their hands. Niels asks if you could explain more about how you image a molecule or a cellular structure that isn't staying in one place.
Right. That's a very good question. Of course, if it's not staying in the place, you have to be fast. You have to get the photons as fast as you can in order to locate it. So the good thing about the min-flux concept is that it requires much fewer photons than any other method in order to find out where the molecule is. So we can be faster. As I explained, seeing the molecules roughly by a factor of 10 or 100. But of course, once it gets very fast, then it's not possible to see the structure anymore because the structure is changing. And then, of course, the limits are reached. So the practical limits are actually given by the fast movements of the molecules. But then you study the movements. But that's also fine.
I'm delighted to tell you here that we have a question from Cynthia Friend, the new president of the Kavli Foundation. So Cynthia, welcome to you and congratulations on your new post. And here's our question for you. You're continuing to push the limits, special resolution and resolving events in time in your more recent work. Can you explain how you're making these advances and how you and your team come up with new ideas?
This is a very good question, actually. I think it's very important how should I put it? I mean, not to work all day and not getting caught by all kind of committees and things. I know maybe I shouldn't say that, but
go ahead. You don't like committees?
Yeah, I agree. Yeah, and sometimes committees do not do good jobs either for fundamental reasons, I think. That's my personal view. So I think it's very important to have enough spare time and to let ideas flow and discuss things with people over coffee and to look at a greater picture. Of course, very important to go into details. In the end, it has to work and you have to put an idea to a test, and nature is, of course, merciless. If something doesn't work, it doesn't work, and there's no way you can convince nature to make it work. But I think it's very, very important to be laid back about something and to let ideas flow and not to be going about after something in a very stiff way. If you do that, probably it's not going to work. That's my take, at least.
Are you laid back?
I think so. Yes, I think so. Yeah.
So what's it like in your lab? I mean, what's it like to be a young scientist working with Stefan Hell?
Well, I think it's actually, personally, I think that it's nice. That's I try to make it as nice as I can. I mean, right out to my door, if you right here, there's a big coffee machine and a place where we gather. And so I don't travel so much, to be honest with you. So I prefer staying here and talking to the students. I don't have a large group anymore. I used to have a larger group in the past, but I like to mingle with students and with postdocs and discuss ideas and see how things go. And I think this is I enjoy it. I must say, I enjoy that very much.
And you have, speaking of time and having fun, you have 4 kids that you're.
That's right. Yes.
So how do you work your very busy science life with your very busy family life?
Actually, yeah, that's a very good question. So we have 4 kids, and a twin boy, 16, a girl, she's 12, and a young boy, he's 6. And so they called me up on my cell phone saying, so when are you coming home? Do we play soccer tonight in the backyard or something. And yeah, that's what I do. Yeah, or go for a hike basically every night.
You go for a hike every night? Wow.
Yeah, virtually every night. Yeah, so at 7 o'clock or so, even with a flashlight or so in the worst case. Yeah, we go for a hike and yeah.
Here's another question from an audience member. Is there a fundamental reason why this technique that you're talking about cannot eventually supply holographic imagery of organism cells in vivo? That's the question.
I mean, it fundamentally relies on using fluorescent molecules. And this is why, say, a pure holographic method that just relies on phase changes wouldn't work. So it's a fluorescence method, and it clearly relies on using fluorescent molecules. But thanks God, say 80% of the life science applications in microscopy and optical microscopy are fluorescence methods. So in that sense, it's not a real restriction, but pure holography in that sense wouldn't work.
Doris asked a question, and probably a lot of folks listening to you and looking at this incredible imagery and the resolution revolution that we've talked about here might have. Doris asked, what insights into biological systems has this technique provided? Answers to questions that could not be answered before?
Yeah, so we are in the beginning of that. I mean, this has been applied in the study of small vesicles, how vesicles fuse, for example, in neurons. It has been applied to finding out about cytoskeleton in neurons, for example. It has been applied to mitochondria, especially the last technique, MinFlux, that I showed was used to optically dissect protein clusters in the so-called cristae of the mitochondria. That's a technical term. And due to the fact that the resolution improvement is so large, of course, I have no doubt that many new discoveries will be made as a result. I think from my perspective, the most important thing right now is to make the technique available so that those who are really specialists in making biological discoveries are in the end capable of doing so.
I'm delighted to say as well, we have a question from the Secretary General of the Norwegian Academy, Gunn Elizabeth Berklund, who asks, you've benefited from finding and being given opportunities to test your ideas, and surely that's come through in the discussion that we've had here, and it's very impressive. But the question goes on, this has been extremely successful, but sometimes research can be risky and the outcomes may not be as expected. Still, we need funding for basic research. Would you comment on this?
Yeah, I think this is a very, very important issue, and honestly, I haven't found a good solution for it, because what I made a remark about committees. So what I learned actually, that committees, when they make a decision on a certain subject or on a certain grant or whatever, or on a certain person, usually they manage to stop the very bad projects, but at the same time, they kill the best projects. I didn't mention it, but the truth is, when I came to this place in Göttingen, I didn't have enough money to begin with to start out this diffraction barrier breaking idea. So I applied for a grant, and it went to a committee with 12 people, and the committee rejected that grant. They rejected that grant outright. And then the officer of that agency decided to give me the money because I complained. And that money, 6 months later, produced the data that was cited in the end by all the committees for the prizes that I got. And so it shows you that the committee of 12 people, of course, didn't realize that I was about to do to make a difference. And so I think committees, that's a big problem, because in committee situations come up where, say sometimes there has to be a consensus on something. And if there is a consensus on something, sometimes the truth is missed out, because you have a consensus and you don't take risks by making sure that there is a consensus.
But what you're saying is that funding is essential. Risk-taking is essential. That's
Absolutely.
What curiosity-driven science is essential, and we need to fund that. But that does, to some extent, certainly here in the United States, get to some issues about public opinion and public support of this kind of funding and all. How do you feel about the sort of assault that many see on science and the questions of trust in science these days? How does that look from where you sit in Germany?
I think, in my view, there is no alternative to doing science. I mean, that's very clear. I mean, humankind has developed in that way that they have tried to better their life, and science is just the result of that. And I think the most ardent problems of humankind can only be solved by science. And of course, there's always people who oppose science for some reason, but in the end, I'm sure there is no alternative. We have to look into cause and effects. And I think what I mean, we as humans, I mean, we stand out perhaps by the fact that we easily can identify cause and effect. And we have to find out what is the cause and what's the effect, and then we can control things and utilize them actually for improving our lives and improving the conditions on Earth. So I think there's no way out there's no alternative to doing science. We have to do science. We have to rely on the scientific procedure.
Here. Which is one of the reasons why we're here, and one of the reasons we thank you for your time and spending this time with us. And I want to come back to your work and a couple of questions from the audience that comes back to some particulars. They're related. Murray asks, could a moving object be tracked with this system? And Jiang Wang asks, can frozen biological samples be imaged to capture images of moving proteins?
Yes, absolutely. Yes, moving molecules can be tracked with higher precision and at higher speed than with any other, say, method. So, with a camera, that has been the normal way of doing that. Can frozen objects be seen? Yes, that's correct. Yes.
And Mary Woolley has a question. Mary asks, speaking of young scientists, we know many of them want to have training in communication and public engagement. Do you think this is a valuable training for academia to offer young scientists? And maybe this is a pitch for you and your donuts here. You
know? No, I think it's very important. I mean, to train the next generation. I mean, it's very clear. And now, having gone through all this hardship in the beginning, I'm asking myself, what can I do now in order to make life easier for the young generation? And I think, from my perspective, we should people allowed to take risk, and we should support them. And of course, we should try to make sure that say taking risk in science doesn't equate taking high social risks. I think this is the problem. So people should be rewarded for taking risks, even if they fail.
Mary also asks about training in communication and public engagement, and this is sort of the communicate, how we communicate science and how we connect these dots for people. So I referenced the donut, because your explanation of the donut,
yeah, right. I mean,
brings us down to things that people can relate to.
That's right. I mean, yes, training, communicating science is, of course, critically important, because after all, science, I mean, unless it comes from a private entity, at least over here, and to a large extent also in the United States, science comes from taxpayers' money. It's paid by taxpayers' money, and if you have to justify why you're doing it. And I think scientific discoveries that really turn out to be very useful, like, for example, the vaccines in the pandemic and so on, they are a clear case for making science, because they show that, yes, science is very useful. I think such occasions should be used in order to promote science and the cause of science.
It also shows that we cannot take for granted that when people are explained the science or shown the facts, that they will accept that. This is an ongoing effort to engage and explain and communicate.
Absolutely. I mean, some people are better at that. I admit some people are not so good at that, but so that shouldn't be decisive on whether somebody makes a career in science. But sure, I mean, it's clearly very helpful to kind of communicate with a larger audience and show them why science is so beneficial.
You are a wonderfully engaging communicator, and you do a great service when you do that. So, in the last question from the audience here, before I ask last one and we turn things back over to Eric, Carol writes, and this is a wonderful question, I think, to wrap up with from the audience. Carol writes, I was interested to hear that no one believed your ideas initially, but you kept going. Did you ever doubt that you were going down the right track?
Good question. The answer is yes. Of course, I doubted at some point, but then the reason why I kept going was very simple. I tried to check the facts. What are the problems that I'm facing? And I realized the problems that I was facing were not conceptual problems. So the physics was correct. So I realized that, and the arguments actually, and the reasons why people came up with say arguments it wouldn't work because of that. They were basically technical. They were not conceptual. They said it's not going to work because the lasers are not there, or because the fluorophores are not there, and this kind of thing. And I realized that these technical problems can be overcome, and they will be overcome with time. Not by myself, but because technology progresses, and so laser technology will progress, and the rest of it, and then it'll become possible at some point. And this was one of the reasons why I kept going. So I felt that there is no fundamental problem. And if there's no fundamental problem, it's likely going to work out. I think for say, any young scientist, it's very important to realize if the person faces a problem, he or she faces a problem, is it a conceptual problem or is it a technical problem? Technical problems will be sorted out over time, I'm sure. Conceptual problems will stay.
Last question to you as we wrap here, and again, thank you for your time and for your insight. What's next in the resolution revolution?
That's a very good question. I mean, the revolution is over, more or less. I mean, it's a very good question. So I'm thinking myself whether I should do something else. Of course, maybe it's time to move on, do something totally different. But I've had a lot of fun with my students in the last, say, 2-3 years, and now we are there. And it's, so invite me in 5 years' time, then we'll see.
Well, and now we'll see the impact that this work that you've had has in the life sciences and other things, because this is when it really will come to life. Thank you so much for your time. This has been so interesting, and I look forward to more, I hope. And Eric Isaacs, let me turn it back over to you with great thanks to Carnegie and to Kavli for making this fabulous discussion possible.
Thank you so much, Frank. That was a great interview. And Stefan, thanks so much for the talk, but also the invention. And really a lot more to ask, but I guess we're out of time. I think you had a lot of great questions here. I also want to thank our audience for asking all these wonderful questions, for joining us in the Kavli Foundation and the Norwegian Embassy as well today for this wonderful lecture. And we look forward to seeing you again in the future. I'd encourage our friends to join us on July 10th for one of our own scientists she's a mineralogist. Her name is Sally Tracy, and she'll give some really interesting information about how minerals tell us about the history of the Earth. So we look forward to seeing you then. And thanks everybody once again. Take care.